Hello? All right, great, it works. All right, hello everybody. Thank you for joining us today in this amazing 2023 iGEM event. This is actually my first time, so I'm super excited to be here with you guys today, and I hope you guys are too. We are the East Coast Bio Crew, a team of high school students based in Baltimore and New York City, and we are here today to introduce you all to our project, RhizeUp, and how soil bacterium can be the key to combating nutrient pollution as well as saving our waterways. There we go, all right. So I think we can start with red tides and dead zones. These two devastating phenomena are the result of eutrophication, an overabundance of nutrients in our waterways. This allows the spur of growth of harmful algae that can deprive an aquatic environment of its resources, such as the sunlight and oxygen, causing possibly permanent damage to the environment. Now, this effect of eutrophication has been in the lives of my teammates and I think, long before our project even began. I, for one, know of a pond right behind my high school, and I knew it was once a thriving ecosystem of lots of creatures, but now it's just smothered with a thick layer of green algae. And this is the effect of eutrophication, and I think that everyone here has seen this before. So that begs the question, where does all this excess nutrient come from? Global modern agricultural practices rely heavily on chemical fertilizers to increase crop output as well as to keep up with ever-growing global food demands. But did you know that more than 80% of the phosphate present in fertilizer never ends up being used? Wow. This is because phosphate readily binds to minerals present in our soil to form a mineral phosphate that's unusable for plants. As a result, the majority of the nutrients we pour into our soil doesn't end up in our plants, but as runoff, reaching our waterways and poisoning our aquatic ecosystems. Now, the effects of eutrophication is on the rise as the use of fertilizers on the rise too. It is estimated that global consumption of fertilizer has surpassed 20 million tons annually. At the same time, cleanup and prevention efforts in the US alone has costed communities more than $1 billion in the past 10 years. And that's just huge numbers. It's terrible. In addition to that, I think the COVID-19 pandemic, as well as the recent conflicts in Europe, has reminded us and emphasized the potential for fertilizer prices to skyrocket and spiral out of control. Just as an example, Russia and its ally Belarus jointly controls more than 18% of phosphate fertilizer trades around the world. We are the East Coast Bio Crew, and our project is RhizeUp. Here at RhizeUp, we are engineering Rhizobium, a soil bacterium known for its symbiosis with legumes, to better solubilize mineral phosphate and to uptake in a form that plants can use to reduce the reliance and use of fertilizers. Now, our implementation plan will involve both stakeholders, such as farmers, and organizations such as the Chesapeake Bay Foundation. Now, for farms, our project can be implemented as seeds inoculated with our genetically modified organism around regions where runoff is particularly of high stress, and that will collect the runoff using our solubilization methods in our bacteria in the roots of the plants, which will uptake it. Now, with this phosphate in our plants, creating a phosphate-rich organic material, we can then reuse this phosphate-rich organic material into a novel biofertilizer to be re-implemented into the farms. Now, we also hope to collaborate with organizations such as the Chesapeake Bay Foundation to introduce similar plants inoculated with our rhizobium in urban areas like storm drains, as well as major bay areas like the Chesapeake Bay or the Charles River. In the long term, our project contributes to a novel regenerative agricultural paradigm in times of geopolitical distress and unknowns.
So now let's go ahead and dive into how we actually engineered our bacteria. First of all, we're introducing the pstSCAB pathway from E. coli. This is a high-affinity phosphate transporter formed by the channel membranes in the periplasm of the bacteria, pstC and pstA. we have pstB as the ATP hydrolyzing protein that's going to help us power this transporter. pstS acts as a protein that binds to the inorganic phosphate and delivers it to the pstABC transporter. The second part is the PQQ-GDH pathway. This will be involved in solubilizing our mineral phosphate. So we have GDH, which is glucose dehydrogenase. This is an enzyme. And we also have the redox cofactor pyrroloquinoline quinone. So PQQ. What this does is it's able to enzymatically convert glucose into gluconic acid. Now, this organic acid has the ability to solubilize phosphate by chelating the heavy metal or mineral that is binded to it, essentially freeing our phosphate and making it available for the plants to use. So, next slide. So, after we used electroporation to integrate our genes into our bacteria, we ran a blue-white screening to test whether or not they were in there. So this is because we used the lac operator. After we confirmed that our genes were inside of our bacteria, we ran a gel electrophoresis to see whether we have, we used the gel electrophoresis to measure the DNA fragment lengths to confirm whether our genes are there. Lastly, we ran a protein gel to see whether or not the proteins we needed were being synthesized. From our protein gel, we have some evidence that our glucose dehydrogenase enzyme was being produced, as you can see by that bar with the arrow. In addition, we ran a series of phosphate assays to see how our rhizobium were uptaking and solubilizing. So we have a series of plate assays that contained media with high tricalcium phosphate. Now, this tricalcium phosphate appears very cloudy. So when our bacteria are uptaking this phosphate, it's going to create a clearing-like halo. As you can see in the images, we see some halos, but still more research and testing needs to be done to confirm these results. In addition, we ran a series of colorimetric assays to test phosphate solubilization. A chromogenic complex would be binded to the free phosphate and turn the media green. Then we could use a spectrophotometer to measure absorbance, which would be directly correlated with the amount of phosphate in the media. We saw that our engineered bacteria had markedly lower absorbances, meaning that there was less phosphate in our media. This is indicative of having our pstSCAB pathway uptake more phosphate, and we also theorized that our PQQ-GDH pathway could be solubilizing the phosphate, and the bacteria are now able to use that for growth, showing why we might have less phosphate in that assay.
For our math modeling subteam, our primary goal was to model the growth of our rhizobium under various conditions, which we did using optical density measurements and sigmoid growth curves. The general equation for a sigmoid growth curve is expressed as followed, with A representing the maximum growth rate,